All right. Hello, everybody. Thank you for coming. My name is Alexander Brinkley, and I am a biology major. And my faculty mentor is Dr. S. Hollis Woodard. And this is quantifying sugar levels in bumblebee hemolymph in relation to sublethal doses of pesticides. So bumblebees are critical pollinators, and they are ideal organisms for the study of energetics. They exist in a constant state of absorbing and breaking down sugars into usable energy, and such they are ideal for studying the chemical process of distribution of sugar throughout the body, sugar and metabolites throughout the body. Sugars from nectar are broken down and transported in the hemolymph. Hemolymph is a fluid contained within the open circulatory system of invertebrates. Imidacloprid is a widely used neonicotinoid pesticide that has been implicated in declining bee populations. It's an acetylcholine receptor blocker, and what it does is at high enough exposure levels, it leads to paralysis and the eventual death of insects. It is also an agonist, so imidacloprid also has sublethal negative effects on bees. So an example of this is it reduces colony health, it has weakened immune systems, and it also leads to abnormal foraging behaviors. So this is the chemical structure of imidacloprid below. Sugars also play a very important role in the central carbon metabolism. The central carbon metabolism is a series of biochemical pathways by which sugars, amino acids, and other biological macromolecules are converted into the precursors of energy metabolism. These precursors include those involved in glycolysis, tricarboxylic acid cycle, and the electron transport chain, all of which are very essential to energy production within bees. They're also needed to meet the bee energy requirements, and they're transported by the hemolymph. Thoracic muscles are very important because they power flight, and they are necessary for foraging, which is flight and foraging are very energetically demanding behaviors displayed in bees. And these muscles hemolymph is in constant circulation and interaction with these tissues and muscles. These studies were designed to examine the effects of imidacloprid on energy metabolism. So for our research questions and the hypothesis, the effects of imidacloprid on bumblebee sugar and metabolite levels are still largely unknown. It is unclear how varying nutritional conditions impact concentration of sugars and metabolites. So I have hypothesized that by exposing bees to sublethal doses of pesticide, it will decrease the concentration of sugars and the relative abundance of metabolites within hemolymph. For the overview of our methods, bumblebee workers were exposed to sublethal doses of imidacloprid valued at 5 parts per billion. This is a field relative dose that is often experienced in the wild. There were 4 treatment groups, each with varying exposure to sublethal doses of imidacloprid, and sugar levels in hemolymph were both quantified and the metabolite abundance in thoracic tissues were analyzed. There were 4 different treatment groups. There's chronic, which is 7-day imidacloprid exposure, acute 1, which is also known as acute early. The first 2 days of the 7-day period, we had imidacloprid exposure followed by 5 days of untreated exposure. Acute 2 or acute late, which was the first 5 days untreated and the last 2 days treated with imidacloprid exposure. And the last day, the last treatment, our untreated group, which was 7 days with no imidacloprid exposure, serving as a negative control. The chronic is pretty clear. We want to see the 7-day exposure of imidacloprid, of sublethal doses of imidacloprid on bumblebee activity and metabolite levels. And acute 1 is for testing the history of exposure, looking at, is there an effect in the first 2 days that is potentially lasting throughout the 7-day period? And for acute late, we wanted to see, are the last 2 days of imidacloprid exposure sufficient enough to see effects in the metabolite levels and concentrations of glucose and overall metabolites within the central carbon metabolome. This is a figure to visualize our previous figure. And this is just the paradigm of treatments showing the 7 days, 5 days, and 2-day treatments of the acute late, acute early, and chronic feeding periods. For our collection methods, following the imidacloprid exposure, bees were briefly starved and then refed with untreated nectar. The purpose of this was to make sure that they were encouraged to feed through the starvation period for 1 hour, and then they were refed in order to make sure that there was sufficient sugar within their systems prior to testing. Hemolymph and tissue were collected at 8-hour intervals within a 24-hour window, and these treatments were at 0 hours, 8 hours, 16 hours, and 24 hours with the respective times next to them. Total for the experiment, we see that there are 177 bees collected across the various treatment paradigms as well as the time collection windows. For the Hemalymp collection, Hemalymp was sourced from the antenna method of Hemalymp extraction. This was optimized in prior research of mine. And the goal of this was to separate the head from the body and extract the hemolymph via the antennae method. And so there you see in the image below, you see that there is a drop of pure hemolymph collected from a bumblebee worker's removed antennae at the head. And bee thoraxes were separated and snap frozen for further metabolomic analysis. This was also important because, as mentioned previously, the flight muscles within the thoraces are very metabolically demanding. And also, so there's lots of presence of metabolites and ATP. For the results of our glucose assay, sublethal imidacloprid exposure potentially produces lower levels of sugars in bumblebee hemolymph. Our preliminary data suggests this. Trends indicate that pesticide treatment does negatively impact the bee sugar levels. However, our findings are inconclusive. There was a very small sample size per treatment group, and this is mostly due to limitations in data analysis due to the COVID-19 pandemic. So this is our figure for the glucose concentration. On the y-axis, we see glucose concentration across treatment at the x. And it's important to note that the untreated group was our significant data showing that untreated groups did experience higher amount of glucose concentrations per pesticide treatments. So that's just something important to note. For the metabolomic analyses, 78 total metabolites were identified, 37 of which were impacted by imidacloprid treatments. We're going to be focusing on 4 metabolites that were impacted within the central carbon metabolome. These include hexose, cysteine sulfonic acid, nicotinamide adenine dinucleotide, also known as NAD, and adenosine diphosphate, also known as ADP. This is our first figure, hexose. Across all of these figures, we see relative abundance of the metabolite on the y-axis, and our x-axis is the sampling time point from 0 to 24 hours. Hexose is a simple sugar. It's a 6-carbon ring structure, and it's essential for energy metabolism. Common representations of hexose include fructose and glucose, which are very common in bumblebee tissues and hemolymph. So something to note, at the start of this 24-hour period, we see that the relative abundance of these metabolites are relatively uniform and at the similar levels. And so it's interesting, though, because the 3 treatment groups in the blue, red, and orange fall off immediately after the 24-hour period, while the untreated group remains relatively stable throughout. So this does help to it's pretty similar to our previous figure with the glucose concentrations, showing that the imidacloprid does in fact impact the relative abundance of hexose, aka glucose, in bumblebee tissues. And potentially these large error bars in the untreated group in green could be due to natural biological variation in the sugar levels of untreated bees, simply due to differences in nectar consumption. Cysteine sulfonic acid, this is a non-coded amino acid. It does play a pretty significant role in amino acid metabolic pathways. It's also important in the taurine metabolism, which is it's a precursor. And so taurine does play a neuromodulatory role in insects. The relative abundance in cysteine sulfonic acid does actually increase over time. If you pay close attention to the orange and the green lines, orange is chronic, green is untreated, we see that they are pretty opposite of each other, with the orange increasing much more, the chronic groups of the chronic treatment showing relative abundance of cysteine sulfonic acid increasing over time, with untreated increasing at a far lower rate. Nicotinamide adenine dinucleotide, NAD, it's an electron carrier involved in the electron transport chain. It's a system for large ATP production, and it plays a very important role in energy metabolism. It's just produces a lot of energy. Looking at this figure, we see the orange and green groups, the chronic and untreated groups, also being of importance. At the 8-hour time point specifically, we see the treatment groups varying quite a bit, with the orange group increasing in the chronic treatment increasing in relative abundance of NAD, and the untreated group decreasing. And these groups eventually pick back up later on and become more similar. A very similar thing is noted here at ADP. ADP is the precursor to ATP, which is one of the fundamental units of cellular energy. It's indicative, ATP and ADP presence is indicative of how much energy is being present or utilized within the organism. At the 8-hour time point, we see a very similar trend as NAD. So sugar levels do tend to decrease as there's an increase in imidacloprid exposure. Key metabolite abundances are impacted by the imidacloprid exposure, examples of which include hexose, cysteine sulfonic acid, NAD, and ADP. However, we cannot make any definite conclusions as analysis is still in process. This work is going to aid in making clear the intersection between pesticide exposure and the functional ability of bees. Our work does help to understand the energetic state of bumblebees and the global decline of bumblebee populations. Future work does include continuing experimental trials and analysis of effects of imidacloprid on metabolites. I'd like to thank my graduate student mentor, Natalie Fisher, and my faculty mentor, Dr. S. Hollis Woodard, our collaborators, Dr. Jay Kirkwood, and my Chancellor's Research Fellowship, which helped offset the costs from 2019-2020. These are the references. And thank you for listening.
Thank you so much, Alexander. That was very